I'm going to share with you my passion for science. I'm not talking about science that takes baby steps. I'm talking about science that takes enormous leaps. I'm talking Darwin. I'm talking Einstein. I'm talking revolutionary science that turns the world on its head. In a moment, I'm going to talk about two ideas that might do this. I say might because with revolutionary ideas, most are flat wrong. And even those that are right seldom have the impact that we want them to have. To explain why I picked two ideas in particular, I'm going to start with a mystery. 1847, Vienna, Austria. Ignaz Semmelweis was a somber, compulsively thorough doctor who ran two maternity clinics. They were identical, except for one thing. Women were dying of high fever soon after giving birth 3 times more often at one of the clinics than at the other. Trying to figure out what the difference was that caused this, Semmelweis looked at everything he could. Sanitation? No. Medical procedures? No. Airflow? No. The puzzle went unsolved until he happened to autopsy a doctor who died of an infected scalpel cut. The doctor's symptoms were identical to those of the mothers who were dying. How is that possible? How could a male doctor get the same thing as new mothers? Semmelweis reconstructed everything the doctor had done right before he got sick, and he discovered that he'd been autopsying a corpse. Had something gotten in his wound that killed him? With growing excitement, Semmelweis looked for any connection he could between dead bodies in the morgue and dead mothers in his delivery room. And he found it. It turned out that at the hospital with a high death rate, but not the others, doctors delivered babies immediately after autopsying corpses in the morgue. Aha! Corpses were contaminating the doctor's hand and killing his mothers. So he ordered the doctors to sterilize their hands, and the deaths stopped. Dr. Ignaz Semmelweis had discovered infectious disease. But the doctors of the day thought he was crazy because they knew, and had for hundreds of years, that odorous vapors called miasmas cause disease, not these hypothetical particles that you couldn't see. It took 20 years for Frenchman Louis Pasteur to prove that Semmelweis was right. Pasteur was an agricultural chemist who tried to figure out why milk and beer spoiled so often. He found the bacteria were the culprits. He also found that bacteria could kill people in exactly the same way that Semmelweis's patients were dying. We now look at what I want to talk about tonight in two ideas. We saw with Semmelweis that he was a revolutionary. He did it for two reasons. One, he opened our eyes to a completely new world. We'd known since the 1680s about bacteria. We just didn't know that bacteria killed people. And he also demolished fond ideas that people kept close to their heart. Miasmas didn't kill people, bacteria killed people. So this brings me to the two ideas I want to talk about tonight. One has opened our eyes to a completely new universe, and the other attacks long-held beliefs. Let's get started with Dr. Eric Betzig. He's a physicist who's opened our eyes to an entirely new world by violating the laws of physics. Betzig is a true rebel. He quit a job at a prestigious Bell Laboratory inventing new microscopes for biology because he thought scientists were taking his brilliant inventions and doing lousy work with them. So he became a house husband. But he never lost his passion for figuring out how to get microscopes to see finer and finer details than had ever been seen before or ever could be seen. This is crucial if we're ever going to understand how cells work and how cancer works and how something 150th the size of the head of a pin can do all these amazing things, like make proteins and move charges around and all of those things. There's just one problem. There's this thing called the law of physics. And part of the law of physics is a thing called the diffraction limit. The diffraction limit is kind of like when you go to a doctor's office, you can only see so far down, no matter how good a glasses you have. This was a so-called impossible problem. But one of Betzig's friends figured out how to take a tiny molecule that was smaller than the best microscope could see and get it to light up and fluoresce. Aha! Betzig said, I think maybe the laws of physics are not so breakable after all. So he lashed together a microscope in his friend's living room. He had no laboratory. This revolutionary instrument got different protein molecules to light up in different colors. And with a computer, he was able to turn very, very fuzzy blurs into very sharp dots and produce images of unprecedented and startling clarity. For this work, last year, Eric Betzig won the Nobel Prize. Why? Because now we can see with unprecedented detail things that we never had seen before, and now doctors can get a better handle on things like cancer. But do you think Betzig was satisfied there? No. He wanted movies. The problem was that even the genius microscopes that he invented were just too slow. So what did he do? He came up with a 200-year-old idea called Moyet patterns. So the way that works is if you take two very, very fine patterns and you move them across each other, you will see a gross pattern that a microscope can see that otherwise you would not be able to see. So he applied this technique to taking a really blurry image of a cell and moving lots of structured light patterns across it until this cell became crystal clear. And here is the result. A mysterious new world, full of strange things zipping around, doing things, and we don't know what they're doing. But when we figure it out, we'll have a better handle on life itself. For example, those green globs that you see, those things are called clathrons. They're molecules that protect other molecules as they move through a cell. Unfortunately, viruses sometimes hijack those to infect cells. Also, you see those little squiggly worm-like things moving around? Those are actin molecules. Unfortunately, viruses also climb down those things to get into the cell nucleus to replicate themselves and make you sick. Now that we can look at movies of what's actually going on deep inside a cell, we have a much better chance of curing viral diseases like AIDS. So when you look at a movie like this, it's very clear that Betzig has opened our eyes to a completely new world. But he hasn't shattered any cherished beliefs. That leads us to Dr. Aubrey de Grey of Cambridge. De Grey definitely has scientists squirming with an interesting idea. We can be immortal. We can beat aging. Now, most scientists think he's a crackpot. Any Biology 101 student knows that aging is an inevitable consequence of living. For example, when we eat, we take in food and we metabolize it, and that throws off what we call free radicals. You might have heard of those, also known as oxygen ions. Those bind to our DNA, cause it to mutate, and cause us to get old and lose our hair. It's just like, no, it's exactly like oxygen binding to iron and making it rust. So you age because you rust out. Okay? Oh, and scientists also know there is something called immortality in cancer cells. So if you stop aging, all of you are going to turn into giant walking malignant tumors. These are cherished beliefs, but could de Grey be onto something? I think he deserves a closer look. First of all, I have a really hard time seeing him as a crackpot. Yeah, he started off life as a computer scientist, not a biologist, but he earned a PhD in biology from Cambridge, and he has published some very significant work on mitochondrial DNA and a bunch of other stuff. Secondly, he started an anti-aging foundation that has identified 7 different causes of aging, to me, that seem very plausible. And he is hot in pursuit of fixes for every single one of them. For example, one of the reasons we age is that our mitochondrial DNA mutates, and we get kind of old and our cells lose energy. He believes, and he's made a convincing case, that using viruses we can do gene therapy, fix that DNA, and rejuvenate our cells. One more thing. We have an existence proof that extreme longevity is possible. Bristlecone pine trees live 5,000 years. And some lobsters don't age at all. Now, this doesn't mean that de Grey is going to revolutionize our lifespans. I mean, after all, we're not trees, and most of us are not lobsters. But I got to believe that there are Darwins and Einsteins out there. And I'll tell you why. Consider this. there are 7 times more people alive today than during Darwin's time. There are 4 times as many people alive today as Einstein. When you consider that the proportion of scientists in the population has skyrocketed, there are now 7 million scientists. I gotta believe, and I do believe, that there's one of them out there who is working right now in obscurity to rock our lives. And I don't know about you, but I can't wait to be rocked. Thank you.
By raising your hand, how many of you know at least one of the person on the screen? Well, it's almost a full house. It's true, they are very famous in their field. And do you know what all of them have in common? They all died of pancreatic cancer. However, it's very, very sad, this news. It's also thanks to their personal story that we have raised the awareness of how lethal this disease can be. It raised to be the 3rd cause of cancer deaths, and only 8% of the patients will survive beyond 5 years. That's a very tiny number, especially if you compare it with breast cancer, where survival rate is almost 90%. So it doesn't really come as a surprise that being diagnosed with pancreatic cancer means facing an almost certain death sentence. What's shocking, though, is that in the last 40 years, this number didn't change a bit, while much more progress has been done for other types of tumor. So how can we make pancreatic cancer treatment more effective? As a biomedical entrepreneur, I like to work on problems that seem impossible, understanding their limitation and trying to find new innovative solutions that can change their outcome. The first bad news with pancreatic cancer is that the pancreas is in the middle of your belly, literally. It's depicted in orange on the screen, but you can barely see it until I remove all of the other organs in front. It's also surrounded by many other vital organs, like the liver, the stomach, the bile duct, and the ability of the tumor to grow into those organs is the reason why pancreatic cancer is one of the most painful tumor types. The hard-to-reach location also prevents the doctor to surgically remove it, as is routinely done for breast cancer, for example. So all of these reasons leaves to pancreatic cancer patients as only option chemotherapy. This brings us to the second bad news. Pancreatic cancer tumors have very few blood vessels. So why should we care about the blood vessel of a tumor? Let's think for a second how chemotherapy works. The drug is injected in the vein and it navigates throughout the body until it reaches the tumor site. It's like driving on a highway trying to reach a destination. But what if your destination doesn't have an exit on the highway? You will never get there. And that's exactly the same problem for chemotherapy and pancreatic cancer. The drugs navigate throughout all of your body. They will reach healthy organs, resulting in high toxic effect for the patients overall, but very little will go to the tumor. Therefore, the efficacy is so limited. To me, it seems very counterintuitive to have a whole body treatment to target a specific organ. However, in the last 40 years, a lot of money, research, and effort has gone towards finding new powerful drugs to treat pancreatic cancer, but nothing has been done in changing the way we deliver them to the patient. So here, after 2 bad news, I'm going to give you a good news, hopefully. With collaborators at MIT and the Massachusetts General Hospital in Boston, we have revolutionized the way we treat cancer by making localized drug delivery a reality. We are basically parachuting you on top of your destination, avoiding you to driving all around the highway. We have embedded the drug into devices that they look like this one. They are flexible enough that they can be folded to fit into the catheter so that the doctor can implant directly on top of the tumor with a minimal invasive surgery. But they are solid enough that once they are positioned on top of the tumor, they will act as a cage. They will actually physically preventing the tumor to enter into other organs, controlling the metastasis. The devices are also biodegradable. That means once in the body, they start dissolving, delivering the drug only locally, slowly, and more effective than what is done with the current whole-body treatment. In preclinical study, we have demonstrated that this localized approach is able to improve of 12 times the response to treatment. So we took a drug that is already known, and by just delivering it locally where is needed the most, we allow a response that is 12 times more powerful, reducing the systemic toxic effect. We are working relentlessly to bring this technology to the next level. We are finalizing the preclinical testing and the animal model required prior to ask FDA for approval of clinical trials. Currently, the majority of the patients will die of pancreatic cancer, and we are hoping that one day we can reduce their pain, extend their life, and potentially making pancreatic cancer a curable disease. By rethinking the way we deliver the drug, we don't only make them more powerful and less toxic, we are also opening the door to find new innovative solutions for almost other impossible problems in pancreatic cancer space and beyond. Thank you very much.
This is a talk about gene drives. I'm going to start by telling you a brief story. 20 years ago, a biologist named Anthony James got obsessed with the idea of making mosquitoes that didn't transmit malaria. It was a great idea and pretty much a complete failure. For one thing, it turned out to be really hard to make a malaria-resistant mosquito. James managed it finally, just a few years ago, by adding some genes that make it impossible for the malaria parasite to survive inside the mosquito. But that just created another problem. Now that you've got a malaria-resistant mosquito, how do you get it to replace all the malaria-carrying mosquitoes? There were a couple options, but plan A was basically to breed up a bunch of the new genetically engineered mosquitoes, release them into the wild, and hope that they pass on their genes. The problem was that you'd have to release literally 10 times the number of native mosquitoes for that to work. So in a village with 10,000 mosquitoes, you'd release an extra 100,000. As you might guess, this was not a very popular strategy with the villagers. Then, last January, Anthony James got an email from a biologist named Ethan Beer. Beer said that he and his grad student, Valentino Gantz, had stumbled on a tool that could not only guarantee that a particular genetic trait would be inherited, but that would spread incredibly quickly. If they were right, it would basically solve the problem that James had been working on for 20 years. As a test, they engineered 2 mosquitoes to carry the anti-malaria gene and also this new tool, a gene drive, which I'll explain in a minute. Finally, they set it up so that any mosquitoes that inherited the anti-malaria gene wouldn't have the usual white eyes, but would instead have red eyes. That was pretty much just for convenience, so they could tell at a glance which was which. So they took their 2 anti-malarial red-eyed mosquitoes, put them in a box with 30 ordinary white-eyed ones, and let them breed. In 2 generations, those had produced 3,800 grandchildren. That is not the surprising part. This is the surprising part. Given that you started with just 2 red-eyed mosquitoes and 30 white-eyed ones, you'd expect mostly white-eyed descendants. Instead, when James opened the box, all 3,800 mosquitoes had red eyes. When I asked Ethan Beer about this moment, he became so excited that he was literally shouting into the phone. That's because getting only red-eyed mosquitoes violates a rule that is the absolute cornerstone of biology, Mendelian genetics. I'll keep this quick, but Mendelian genetics says that when a male and female mate, their baby inherits half of its DNA from each parent. So if our original mosquito was AA and our new mosquito was AB, where B is the anti-malaria gene, the baby should come out in 4 permutations: AA, AB, AA, BA. Instead, with a new gene drive, they all came out AB. Biologically, that shouldn't even be possible. So what happened? The first thing that happened was the arrival of a gene editing tool known as CRISPR in 2012. Many of you have probably heard about CRISPR, so I'll just say briefly that CRISPR is a tool that allows researchers to edit genes very precisely and easily and quickly. It does this by harnessing a mechanism that already existed in bacteria. Basically, there's a protein that acts like a scissors and cuts the DNA, and there's an RNA molecule that directs the scissors to any point on the genome you want. The result is basically a word processor for genes. You can take an entire gene out, put one in, or even edit just a single letter within a gene. And you can do it in nearly any species. Okay, remember how I said that gene drives originally had two problems? The first was that it was hard to engineer a mosquito to be malaria resistant. That's basically gone now, thanks to CRISPR. But the other problem was logistical. How do you get your trait to spread? This is where it gets clever. A couple years ago, a biologist at Harvard named Kevin Esvelt wondered what would happen if you made it so that CRISPR inserted not only your new gene, but also the machinery that does the cutting and pasting. In other words, what if CRISPR also copied and pasted itself? You'd end up with a perpetual motion machine for gene editing. And that's exactly what happened. This CRISPR gene drive that Esvelt created not only guarantees that a trait will get passed on, but if it's used in the germline cells, it will automatically copy and paste your new gene into both chromosomes of every single individual. It's like a global search and replace. Or in science terms, it makes a heterozygous trait homozygous. So, what does this mean? For one thing, it means that we have a very powerful, but also somewhat alarming new tool. Up until now, the fact that gene drives didn't work very well was actually kind of a relief. Normally, when we mess around with an organism's genes, we make that thing less evolutionarily fit. So biologists can make all the mutant fruit flies they want without worrying about it. If some escape, natural selection just takes care of them. What's remarkable and powerful and frightening about gene drives is that that will no longer be true. Assuming that your trait does not have a big evolutionary handicap, like a mosquito that can't fly, the CRISPR-based gene drive will spread the change relentlessly until it is in every single individual in the population. Now, it isn't easy to make a gene drive that works that well, but James and Esvelt think that we can. The good news is that this opens the door to some remarkable things. If you put an anti-malarial gene drive in just 1% of Anopheles mosquitoes, the species that transmits malaria, researchers estimate that it would spread to the entire population in a year. So in a year, you could virtually eliminate malaria. In practice, we're still a few years out from being able to do that, but still, 1,000 children a day die of malaria. In a year, that number could be almost zero. The same goes for dengue fever, chikungunya, yellow fever. And it gets better. Say you want to get rid of an invasive species, like get Asian carp out of the Great Lakes. All you have to do is release a gene drive that makes the fish produce only male offspring. In a few generations, there'll be no females left. No more carp. In theory, this means we could restore hundreds of native species that have been pushed to the brink. Okay, that's the good news. This is the bad news. Gene drives are so effective that even an accidental release could change an entire species, and often very quickly. Anthony James took good precautions. He bred his mosquitoes in a biocontainment lab, and he also used a species that's not native to the US, so that even if some did escape, they'd just die off. There'd be nothing for them to mate with. But it's also true that if a dozen Asian carp with the all-male gene drive accidentally got carried from the Great Lakes back to Asia, they could potentially wipe out the native Asian carp population. And that's not so unlikely, given how connected our world is. In fact, it's why we have an invasive species problem. And that's fish. Things like mosquitoes and fruit flies, there's literally no way to contain them. They cross borders and oceans all the time. Okay, the other piece of bad news is that a gene drive might not stay confined to what we call the target species. That's because of gene flow, which is basically a fancy way of saying that neighboring species sometimes interbreed. If that happens, it's possible a gene drive could cross over, like Asian carp could infect some other kind of carp. That's not so bad if your drive just promotes a trait, like eye color. In fact, there's a decent chance that we'll see a wave of very weird fruit flies in the near future. But it could be a disaster if your drive is designed to eliminate the species entirely. The last worrisome thing is that the technology to do this, to genetically engineer an organism and include a gene drive, is something that basically any lab in the world can do. An undergraduate can do it. A talented high schooler with some equipment can do it. Now, I'm guessing that this sounds terrifying. Interestingly, though, nearly every scientist I talked to seemed to think that gene drives were not actually that frightening or dangerous. Partly that's because they believe that scientists will be very cautious and responsible about using them. So far, that's been true. But gene drives also have some actual limitations. So for one thing, they work only in sexually reproducing species, so thank goodness, they can't be used to engineer viruses or bacteria. Also, the trait spreads only with each successive generation. So changing or eliminating a population is practical only if that species has a fast reproductive cycle, like insects or maybe small vertebrates like mice and fish. In elephants or people, it would take centuries for a trait to spread widely enough to matter. Also, even with CRISPR, it's not that easy to engineer a truly devastating trait. Say you wanted to make a fruit fly that feeds on ordinary fruit instead of rotting fruit with the aim of sabotaging American agriculture. First, you'd have to figure out which genes control what the fruit fly wants to eat, which is already a very long and complicated project. Then you'd have to alter those genes to change the fly's behavior to wherever you want it to be, which is an even longer and more complicated project. And it might not even work because the genes that control behavior are complex. So if you're a terrorist and have to choose between starting a grueling basic research program that will require years of meticulous lab work and still might not pan out, or just blowing stuff up, you'll probably choose the latter. This is especially true because, at least in theory, it should be pretty easy to build what's called a reversal drive. That's one that basically overwrites the change made by the first gene drive. So if you don't like the effects of a change, you can just release a second drive that will cancel it out, at least in theory. Okay, so where does this leave us? We now have the ability to change entire species at will. Should we? Are we gods now? I'm not sure I'd say that, but I would say this. First, some very smart people are even now debating how to regulate gene drives. At the same time, some other very smart people are working hard to create safeguards, like gene drives that self-regulate or peter out after a few generations. That's great. But this technology still requires a conversation. And given the nature of gene drives, that conversation has to be global. What if Kenya wants to use a drive, but Tanzania doesn't? Who decides whether to release a gene drive that can fly? I don't have the answer to that question. All we can do going forward, I think, is talk honestly about the risks and benefits, and take responsibility for our choices. By that I mean not just the choice to use a gene drive, but also the choice not to use one. Humans have a tendency to assume that the safest option is to preserve the status quo. But that's not always the case. Gene drives have risks, and those need to be discussed. But malaria exists now and kills 1,000 people a day. To combat it, we spray pesticides that do grave damage to other species, including amphibians and birds. So when you hear about gene drives in the coming months, and trust me, you will be hearing about them, remember that. It can be frightening to act, but sometimes not acting is worse.
That used to house one transistor, we can now fit 1 billion. That made it so that a computer the size of an entire room now fits in your pocket. You might say the future is small. As an engineer, I'm inspired by this miniaturization revolution in computers. As a physician, I wonder whether we could use it to reduce the number of lives lost due to one of the fastest growing diseases on Earth, cancer. Now, when I say that, what most people hear me say is that we're working on curing cancer. And we are. But it turns out that there's an incredible opportunity to save lives through the early detection and prevention of cancer. Worldwide, over 2/3 of deaths due to cancer are fully preventable using methods that we already have in hand today. Things like vaccination, timely screening, and of course, stopping smoking. But even with the best tools and technologies that we have today, some tumors can't be detected until 10 years until after they've started growing, when they are 50 million cancer cells strong. What if we had better technologies to detect some of these more deadly cancers sooner, when they could be removed, when they were just getting started? Let me tell you about how miniaturization might get us there. This is a microscope in a typical lab that a pathologist would use for looking at a tissue specimen like a biopsy or a pap smear. This $7,000 microscope would be used by somebody with years of specialized training to spot cancer cells. This is an image from a colleague of mine at Rice University, Rebecca Richards-Cordum. What she and her team have done is miniaturize that whole microscope into this $10 part, and it fits on the end of an optical fiber. Now, what that means is instead of taking a sample from a patient and sending it to the microscope, you can bring the microscope to the patient. And then, instead of requiring a specialist to look at the images, you can train the computer to score normal versus cancerous cells. Now, this is important because what they found working in rural communities is that even when they have a mobile screening van that can go out into the community and perform exams and collect samples and send them to the central hospital for analysis, that days later, women get a call with an abnormal test result and they're asked to come in. Fully half of them don't turn up because they can't afford the trip. Now, with the integrated microscope and computer analysis, Rebecca and her colleagues have been able to create a van that has both a diagnostic setup and a treatment setup. And what that means is that they can do a diagnosis and perform therapy on the spot, so no one is lost to follow-up. That's just one example of how miniaturization can save lives. Now, as engineers, we think of this as straight-up miniaturization. You took a big thing and you made it little. But what I told you before about computers was that they transformed our lives when they became small enough for us to take them everywhere. So what is the transformational equivalent like that in medicine? Well, what if you had a detector that was so small that it could circulate in your body, find the tumor all by itself, and send a signal to the outside world? It sounds a little bit like science fiction, but actually, nanotechnology allows us to do just that. Nanotechnology allows us to shrink the parts that make up the detector from the width of a human hair, which is 100 microns, to 1,000 times smaller, which is 100 nanometers. And that has profound implications. It turns out that materials actually change their properties at the nanoscale. You take a common material like gold, and you grind it into dust, into gold nanoparticles, and it changes from looking gold to looking red. If you take a more exotic material, like cadmium selenide, forms a big black crystal. If you make nanocrystals out of this material and you put it in a liquid and you shine light on it, they glow. And they glow blue, green, yellow, orange, red, depending only on their size. It's wild. Can you imagine an object like that in the macro world? It would be like all the denim jeans in your closet are all made of cotton, but they are different colors depending only on their size. So as a physician, what's just as interesting to me is that it's not just the color of materials that changes at the nanoscale. The way they travel in your body also changes. And this is the kind of observation that we're going to use to make a better cancer detector. So let me show you what I mean. This is a blood vessel in the body. Surrounding the blood vessel is a tumor. We're going to inject nanoparticles into the blood vessel and watch how they travel from the bloodstream into the tumor. Now, it turns out that the blood vessels of many tumors are leaky, and so that nanoparticles can leak out from the bloodstream into the tumor. Whether they leak out depends on their size. So in this image, the smaller 100 nanometer blue nanoparticles are leaking out, and the larger 500 nanometer red nanoparticles are stuck in the bloodstream. So that means as an engineer, depending on how big or small I make a material, I can change where it goes in your body. In my lab, we recently made a cancer nanodetector that is so small that it could travel into the tumor and look for tumors. We designed it to listen for tumor invasion, the orchestra of chemical signals that tumors need to make to spread. For a tumor to break out of the tissue that it's born in, it has to make chemicals called enzymes to chew through the scaffolding of tissues. We designed these nanoparticles to be activated by these enzymes. One enzyme can activate 1,000 of these chemical reactions in an hour. Now, in engineering, we call that 1 to 1,000 ratio a form of amplification, and it makes something ultra-sensitive. So we've made an ultra-sensitive cancer detector. Okay, but how do I get this activated signal to the outside world where I can act on it? For this, we're going to use one more piece of nanoscale biology, and that has to do with the kidney. So the kidney is a filter. Its job is to filter out the blood and put wastes into the urine. It turns out that what the kidney filters is also dependent on size. So in this image, what you can see is that everything smaller than 5 nanometers is going from the blood, through the kidney, into the urine, and everything else that's bigger is retained. Okay, so if I make a 100 nanometer cancer detector, I inject it in the bloodstream, it can leak into the tumor where it's activated by tumor enzymes to release a small signal that is small enough to be filtered out of the kidney and put into the urine. I have a signal in the outside world that I can detect. Okay, but there's one more problem. This is a tiny little signal, so how do I detect it? Well, the signal is just a molecule. There are molecules that we designed as engineers. They're completely synthetic, and we can design them so that they are compatible with our tool of choice. If we want to use a really sensitive, fancy instrument called a mass spectrometer, then we make a molecule with a unique mass. Or maybe we want to make something that's more inexpensive and portable. Then we make molecules that we can trap on paper, like a pregnancy test. In fact, there's a whole world of paper tests that are becoming available in a field called paper diagnostics. All right, where are we going with this? What I'm going to tell you next, as a lifelong researcher, represents a dream of mine. I can't say that it's a promise, it's a dream, but I think we all have to have dreams to keep us pushing forward, even, and maybe especially, cancer researchers. I'm going to tell you what I hope will happen with my technology, that my team and I will put our heart and souls into making a reality. Okay, here goes. I dream that one day, instead of going into an expensive screening facility to get a colonoscopy or a mammogram or a pap smear, that you could get a shot, wait an hour, and do a urine test on a paper strip. I imagine that this could even happen without the need for steady electricity or a medical professional in the room. Maybe they could be far away and connected only by the image on a smartphone. Now, I know this sounds like a dream, but in the lab, we already have this working in mice, where it works better than existing methods for the detection of lung, colon, and ovarian cancer. And I hope that what this means is that one day, we can detect tumors in patients sooner than 10 years after they've started growing, in all walks of life, all around the globe, and that this would lead to earlier treatments, and that we could save more lives than we can today with early detection. Thank you.